Welcome to our video. Today we will be presenting Apoptosis.
Apoptosis, or programmed cell death, is a process that occurs in multicellular organisms. It consists of several biochemical events that cause characteristic changes in the morphology of the cell, which lead to its death. These changes include bleeding, cell shrinkage, nuclear fragmentation, chromatin condensation, chromosomal DNA fragmentation, and global messenger RNA decay.
Some 50 to 70 billion cells die each day due to apoptosis in the average human adult. For an average child between the age of 8 and 14, approximately 20 billion to 30 billion cells die a day. Apoptosis is a highly regulated and controlled process that confers advantages during an organism's life cycle. For example, the separation of fingers and toes in the developing human embryo occurs because cells between the digits undergo apoptosis.
Did you know that if apoptosis didn't exist, we would actually look like a ball?
Tissue damage. When the cell is considerably damaged, it has no possibilities of being repaired. This is the case of viral infections. Releasing the contents of the cell would be harmful for the surrounding cells, so it must be eliminated by apoptosis.
Immune system regulation. Occasionally, some cells from the immune system, namely lymphocytes B and T, can develop a tendency to attack healthy cells and tissues from the same organism to which they belong.
Run.
These autoreactive cells are eliminated by means of apoptosis.
Development and homeostasis. Programmed cell death is an essential process in the development of new tissues and structures. When cells die, their components are phagocytosed and reused by macrophages and surrounding cells. Furthermore, the elimination of excess cells and other cells which risk the integrity of the organism is vital for the survival of the individual. In an adult organism, the quantity of cells that make up a tissue or an organ must remain constant, that is, within certain limits. Blood cell and skin cells, for instance, are constantly being produced and renewed by their respective parent cells. Old cells must therefore be eliminated to compensate the proliferation of new cells. But how does apoptosis work? Well, it can follow two main pathways.
Each pathway induces cell death by activating caspases, which are proteases or enzymes that degrade proteins. Once initiator caspases are activated, they activate executioner caspases, which then kill the cell by degrading proteins indiscriminately, breaking the cell into smaller fragments called apoptotic bodies, which can be rapidly engulfed and digested by phagocytic cells. These prevent the contents of the cell from spilling out onto neighboring cells and causing them damage. Because of the cascade reactions it contains, it cannot stop once it has begun, so it is a highly regulated process.
Now we are going to talk about apoptotic phases. The first one is the detection phase. As it was previously explained, it can consist of two different pathways depending on the factors involved. One of them, the intrinsic pathway, is activated by intracellular signals generated when cells are stressed and depends on the release of protein from the intermembrane space of the mitochondria. Since aerobic respiration stops in the mitochondria, the cell runs out of energy, which is ATP, and dies.
The extrinsic pathway is activated by extracellular ligands from neighboring cells, which bind to the cell surface death receptors, leading to the formation of the death-inducing signaling complex.
Decision phase. When the cell receives a death signal, it must decide whether to initialize cell death or survive. The role of the mitochondria is highly important in this process, since it contains a chemical factor which can unchain the reactions that lead to the completion of apoptosis. This factor, which belongs to the family of the caspases, induces apoptosis and is contained in the mitochondrial cytoplasm. If a multiprotein complex, which modifies the permeability of the mitochondrial membranes, is produced, intramitochondrial content, including cytochrome C, is released, triggering the reactions taking place in the execution phase. Other relevant events that happen due to the permeabilization of the membranes are the altering of the electron transporting chain, the loss of the electrochemical potential, and several changes in the metabolic cycle of oxidation reduction.
Execution phase. Once the apoptotic process starts, that is, the cell decides to die, several biochemical pathways which lead to the degradation of proteins and chromatin take place. The main proteases implied in these processes are caspases, calpains, granzyme B, and a multiprotein complex known as proteasome.
Faults in apoptosis. Exaggerated apoptosis can lead to atrophy and organ functional failure. Cancer. When the capacity of a cell to carry out apoptosis is damaged, perhaps due to the mutation, or the initial metabolic reactions of this process have been blocked, for instance due to a virus, the damaged cell could continue dividing without restrictions, resulting in a tumor, which can then develop into a cancer.
That is all for our video. Thank you for your attention. We hope you enjoyed it and it helped you better understand apoptosis.
Vale, 3, 2, 1, referente. Otra. 3, 2. Es que lo hacéis mal.
Ahora hacer la ola, así. Espera,
¿Qué?
espera. Pero parece que se parece más. ¿Para las manos?
A ver, no salgáis del plano tampoco. Vale.
Venga, que viene, que viene, que viene. Que va, que va, que va, que va. Vale, ahora otra para que no se... No.
Os ha jodido un poco la toma, pero...
Hay que hacer otra, hacemos otra y cuando la cambiemos de antes... Vale, tú dices 3, 2, 1 y la echamos.
Va, ya. Quítamelo.